Hi guys, my name's Miss White and I'm here to go over Chapter 1, Big Ideas with you. Chapter 1 is all about the science of biology. So 3 big things to remember is that scientists have an ongoing goal to explain things in our world. Science can be applied to any question where the answer can be observed, researched, investigated, or measured, or in other words, where evidence can actually be found. And something to keep in mind is that science cannot answer questions if evidence is not available. So for example, we can't answer the question, what is the purpose of life? Because we can't actually conduct valid experiments that have valid data. Section 1.1 is about science, and specifically what is science and the nature of science. So remember that science uses evidence to construct testable explanations and predictions of natural phenomenon. What that means is that we need to actually concretely be able to do an experiment to then come up with some sort of conclusion. So I can test to see if a certain light helps plants grow better. I can't actually test to see if there are ghosts that are real. Okay? Science also does generate knowledge. That's a huge thing from this. I'm rarely trying to prove anything because we are actually continually gathering more information and more evidence, and so things actually just naturally shift. That leads us to say that science is a process. It's something that you do rather than just a group of facts. Science also does allow us to make useful predictions about natural events, and we use data to understand patterns within nature. Now, something to kind of mention is that this has changed quite dramatically within the last few years. We want to remember that oftentimes people think that there has to be a certain process that is done when you're studying science, and that's not actually accurate. So when we talk about the scientific methodology, meaning the steps that we take when we are conducting scientific experiments, we want to remember that it's really just a general style of investigation. It's not a rigid process. So although you might have been told that you have to do certain steps in certain orders, just keep in mind it's actually just a suggestion. Scientists use the steps, but sometimes they make it their own, and they may do things out of what we consider to be the regular order. We want to remember that no matter what branch of science you are studying, that you still actually will use a methodology that involves things like coming up with questions, figuring out the purposes, figuring out your materials, etc. So if we look below, there are 2 sets of graphs, location A versus location B. And I can describe the difference between the grasses of these 2 locations. So for example, the grasses in location B are taller than those in location A. So the first thing I notice is that there actually is a difference between these 2 grasses, and so that might actually lead me, based on that observation of saying that one is taller than the other, well, why do marsh grasses grow to different heights in different places? So really taking in your environment and observing things can lead to you coming up with questions that you could then start the scientific process with and then try to answer. Now, observing and asking questions, huge part of science. All scientific investigations involve asking questions. And to answer questions, we try to focus on one specific factor. So why study only one factor at a time, you might be asking? Well, it makes it impossible to tell if observed changes are from one factor or from another. Okay, so if I am changing more than one thing at a time, I can't tell if my outcome is based on the first thing that I changed or the second thing that I've changed. So if a scientist were to study how both the amount of light and the amount of water affected plant growth at the same time, what would the observations and data show, right? We want to keep in mind that it would be impossible to tell if changes in the plants were due to light or water differences if we actually tested for both. Now, we've already talked a little bit about an inference, but I just want you to remember that an inference is just a logical interpretation based on what scientists already know or what you already know. A hypothesis is different than an inference because it's actually a tentative explanation that can be tested further. Okay, so in an inference, I am making an assumption. A hypothesis, I've now taken that and added essentially an educated guess or explanation. So a scientist might make an inference based on that prior knowledge and use that to generate the hypothesis. I might say something like, I'm inferring that there's something with the soil that might be causing some sort of change between location A versus location B. And my hypothesis might be, if soil is affecting the growth rate of location B grass, then changing the pH might cause a dramatic decrease in the height of location B. So I took my inference and then I applied some of my knowledge to figure out how I could actually test for this. Now remember that I do need to design a controlled experiment, and so that means that I am going to have to pay attention to my variables. So in an experiment, there are always two groups. Both groups, the control and the experimental group, have to have the same variables except for one. Remember, when you change more than one variable, you will not be able to concretely say this variable was the cause of the change. Now, we keep a control group, which means that is the group that is completely normal, we don't mess with it. And then the experimental group is the group where I am actually changing some sort of variable. Now let's get a little more technical. What variable changed in the experiment? And that is called the independent variable. So the thing that I am going to manipulate, think about that I in independent. I am going to manipulate is what is going to possibly cause a change. In our experiment on the right-hand side here, you see that we are adding nitrogen to the soil, and that group has the independent variable. Okay. Now the group without it is known as the control group, and that group, again, you leave completely alone. What change occurred due to the independent variable? Okay. Now that, in this case, is the height, the growth rate of the grass. Okay. So all other variables should be the same, and in a controlled experiment, there only is one variable where you have a difference between the two. So in this example here, I have an independent variable that I am manipulating, and the dependent variable, which is the height of the grass, will essentially change based on whatever I have manipulated. Now, there's also different types of data that we can collect. So we have quantitative data and qualitative data. Quantitative with an N, so think about that N in quantitative. That involves numbers and measurements. So this is data where you are measuring things, or you have a certain number of things. Height, for example, is quantitative. If I cannot associate a number to the data, then we would call that qualitative, where you describe things with words. Oftentimes, observations count as qualitative data. So remember, N for number, quantitative data has numbers. Now, what type of data did scientists collect in our experiment? Well, the quantitative data in this experiment with the grass was the height of the grass in the control group and in the experimental group. Remember to list examples of qualitative data, and for our example here, we have qualitative being the grass color, the presence of a plant disease on some leaves, all the things that I can observe. The quantitative data from this specific example would be the number of plants per plot, the length, the width, the height, the weight of each blade of grass, so all the things that I can measure. Now I'm going to go ahead and analyze my conclusions, and those are typically going to be involving data of some kind. So reading graphs and patterns in data is called analyzing data. Based on the analysis, scientists can make a conclusion for that particular experiment. So what is the graph showing, and what can we conclude? Well, in this graph, we can see that the experimental group, which got the independent variable of the extra nitrogen, grew taller than the control group, which did not get any nitrogen at all. So the nitrogen, based on our experiment, seemed to cause the grass to grow higher. So how could the experiment in our example be refined or expanded? And we could actually repeat this, which we would want to do, and then we could also adjust the amount of nitrogen. We could put more nitrogen or less nitrogen, or even use a different type of soil nutrient to see if it would affect the height as well. Here's some general vocab. I would suggest either screenshotting this or going back in and adding these to your notes. You should know that observations are using your senses. Inferences are taking logical interpretations of those observations. A hypothesis is a tentative scientific explanation that you can test. It must be testable. A controlled experiment is one where only one variable is changed. Don't forget, controlled experiment is the name of the whole experiment. A control group is not the same thing, so please don't mix those two terms up. An independent variable is the variable that is being changed or manipulated. So I am changing this, which then will affect my dependent variable. That's the variable that I am observing and that is going to react to whatever I have done. Okay, so for example, does light affect plant growth? The independent variable would be the amount of light I expose plants to. The dependent variable is what happens, which is to see how high those plants grow. Now remember, within a controlled experiment, you're going to have an experimental group and a control group. That control group is the one that is just normal, completely left alone, and you use control groups to make sure that there is some sort of change. All right, so now let's talk about a theory, because oftentimes outside of a scientific classroom or a scientific application, people don't use theory correctly. In the science world, a theory applies to a tested, highly reliable scientific explanation of events in the natural world that unifies many repeated observations and incorporates durable, well-supported hypotheses that enable scientists to make accurate predictions. Okay? So when the word theory is used in science, it refers to an idea that has been thoroughly tested and is supported by a great ideal of evidence, such as the theory of gravity. Now, some people think that theories become laws. They are not the same thing at all. Okay? A law describes behavior. A theory explains why it is behaving that way. Okay, so we say the theory of evolution. We don't say the law of evolution, because laws describe how something is happening. So if you ever look up any different type of scientific laws, you'll notice all they are doing is telling you what is happening, but they're not telling you why it's happening. So just remember, theories do not become laws. And when in science we talk about a theory, we're talking about something that isn't just like, oh, I think this has been repeated over and over and over. And up to that point, there have been no types of evidence that could disprove it. Theories can change though, and that's actually a great thing, because as we get more information and more data, we know that naturally we can update information, so they're not set in stone. At this point in chapter 1.1, you should be able to answer the following. What are the goals of science? What procedures are at the core of the scientific methodology process? And what is scientific theory? Continue on if you want to see chapter 1.2. Otherwise, take a break here. Chapter 1.2, Science in Context. This is all about the process of science. And science, something to keep in mind is that it is dynamic. It is flexible and unpredictable. And what might be a useful quality in a scientist? So we're going to talk about all of that. Now, first thing is exploration and discovery. So ideas in science can arise in many different ways, from simply being curious to the need to actually solve a particular problem. What present-day issue or problem poses a question scientists could address? Well, how to reverse climate change. Oftentimes that is a huge news right now, but that is oftentimes something that is talked about. You could also try to figure out how to clean up pollution problems, how to tackle childhood obesity, and at the moment, very relevant, is how to figure out a proper way to create a vaccine for COVID-19. Technology does provide us new opportunities to ask questions and to gather new forms of data with greater accuracy and quantity. Remember that the technologies developed by scientists have proven to help society tremendously. So, first thing we want to kind of mention on here, and you may want to look through the slides in more depth through the PowerPoint itself, is there's a lot of great visuals on here that show those connections. You'll see the same visuals within your textbook. So do make sure that you look through all of these diagrams. They help connect all of these dots. All right, scientists communicate and keep information objective through peer review. What that means is that they don't just post information to Twitter or Instagram or make a TikTok about it. Scientists actually have to present their information and then have that information criticized, and that is a good thing, okay? We want to remember that published articles in peer-reviewed scientific journals allow researchers to share ideas and allows other scientists to evaluate and test the data and analysis. So when a scientific paper is published, does that mean that research about that topic is complete? Why or why not? Well, publication of a paper does not mean that research about a topic is complete. It may open doors for many new studies about the same topic. If anyone has ever submitted a peer-reviewed article, they know that they are going to get a ton of criticism, and that is a good thing. They want somebody else to validate their information, or if it doesn't actually allow validation, meaning I can't repeat whatever they're trying to do with their experiment, then that tells the scientist that something is very wrong. You either didn't present that information correctly, or it's fake. And so that does happen on occasion. Now, any scientist claiming to have discovered something new must be willing to reveal all of the details for peers to scrutinize and test for themselves. You can't claim you have a COVID-19 vaccine and then not be willing to share that information, because you have to actually have credibility, and that means you have to be able to repeat something over and over and over. And not just you, anybody should be able to repeat that experiment. This all allows us to have a robust bank of knowledge that is credible. Now, how does science influence society? Well, we know that scientific data helps provide answers to questions that affect everyday life. Scientists do not work in a vacuum, meaning they're not by themselves in a lab. Their research is strongly influenced by society. So how does that actually connect together? Well, society can limit the application of scientific ideas, especially if new scientific ideas conflict with prevailing cultural beliefs. So for example, it used to be legal to smoke cigarettes in public places, including restaurants. My parents owned a restaurant, and growing up, there was a whole side of the restaurant, literally 3 feet away from other people, that was called the smoking section, and you could smoke and eat your dinner. And then 3 feet away, there was the non-smoking section. Well, as scientific studies proved that secondhand smoke was dangerous, people started to see cigarettes differently. Eventually, laws changed, banning cigarettes from most public indoor areas, and my parents did that as well. They banned cigarette smoke from the inside. So as more information comes out, and as the societal beliefs and pressures coming down from politics change, we oftentimes see science being applied in different ways. Lastly, we have our science and engineering practices. So although some of the specifics vary, the steps in scientific inquiry and in engineering design are basically the same. You're going to be asking questions and defining problems. You're going to be developing and using models. You're going to be planning and carrying out investigations. You're going to be analyzing and interpreting data. You're going to use mathematics and computational thinking. You're going to construct explanations and design solutions. You're going to be engaging in argument for evidence, and you're going to be obtaining, evaluating, and communicating information. By this point in section 1.2, you should be able to answer the following questions. What attitudes and experiences generate new ideas? Why is peer review so important? What is the relationship between science and society? What practices are shared by science and engineering? If you are all done or need a break, now would be the time to do that. And I'm going to be moving on to section 1.3, so feel free to stop or pause at this point. Chapter 1.3, Patterns of Life. Let's talk about the 8 characteristics of living things. Number 1, they are made up of cells. They can be unicellular, meaning one cell, like an amoeba, or multicellular, meaning millions to billions of cells, like you or me. They must reproduce, which can be asexually, meaning that they make a copy of themselves and then split. There's also a few other ways that we can have asexual reproduction done, but essentially it just means that there is no partner. It's just that 100% set of DNA coming from just the one parent. Or sexual, which simply means that you're taking 2 different sets of genetics and mixing them together. Just as a note, sexual reproduction doesn't actually mean sex, and we'll get into this later when we talk about genetics and evolution. All it implies is a sperm and an egg meeting up and then creating another being from that. Number 3, they are based on a universal genetic code, and that genetic code we call DNA. Number 4, they grow and develop. Number 5, they obtain and use materials and energy. Number 6, they respond to their environment. Number 7, they maintain an internal balance. We call that homeostasis. That means that your things like your blood pressure, for example, your temperature, those things are maintained throughout the body or the system. Number 8, they change over time, meaning that they evolve. Number 8 is the only one that you would not actually be able to directly observe, because in order for something to evolve, it would actually take many, many, many generations. And for things that live a very long time, that would mean millions and millions of years. Okay, so number 8 does need to be there, but that is only something you can tell over a very long period of time. Now, as we look at number 6, oftentimes people think of plants or animals. Sorry, they think of plants and not animals, and they start to question, well, can a plant even really move? Can it really respond? And so you'll notice that the way that this is worded is very specific to not say movement, but response. And so I'm going to show you guys some quick video clips so that you can see that there are plants that do move quite a bit. All plants have the ability to move. They actually will go towards sunlight. However, that movement is so slow to our eyes that we oftentimes don't see them. So stick around, take one moment, watch these videos right now. So you guys were able to see the 3 videos. One of those plants is called the mimosa. That was the one that was closing once it was touched. We actually would normally play with these in class, and you have to be very gentle with them, but all it takes is a very light tap, and then they close off their leaves. We also saw a vine plant that over a number of days was actually able to keep moving until it eventually found something to climb up. And then the last one we saw was a bunch of bean sprouts, essentially, that were moving based on where the light was coming out. And so we can notice this outside in nature quite a bit if you really look for it. So for example, trees that are leaning to the sides, plants that are growing out of the side, they're all moving towards where the sun is. Now, this section is called cross-cutting concepts. And sorry, not section, my slide is called cross-cutting concepts in biology. And these are the huge concepts that we're going to see throughout our whole textbook. A big one is cause and effect. We're going to be seeing a lot of different explanations about what's causing something and then what is ultimately happening because of that. We know that as the mechanism and the explanation. We're going to see a lot of systems and system models throughout our textbook. We're going to talk about stability and change. You're going to notice that patterns pops up a lot. There's also scale, proportions, and quantity. Energy and matter comes up quite a bit, where we talk about different types of flows, of cycles, and of conservation. And lastly, structure and function. Now, there are lots of different fields within biology, and they're all actually interconnected with each other. Global ecology is one, biotechnology is another, entomology is another, and then molecular biology is another big one. Okay. Now, entomology is ecology and evolution of infectious diseases, and molecular biology is really the genes and all of the different molecules that are associated with that. As a quick review about the scientific method, we're going to have some practice with this. So for right now, I just want to mention it. And if you feel like you need more practice, I'll make a separate video on that. We use the metric system, and the metric system uses different measurements than us. They have 3 base units: the meter, the liter, and the gram. And so when we're measuring length, we're going to be using meters. When we're measuring mass, we're going to be using grams. And when we're measuring volume, we're going to be using liters. Within the metric system, they also use Celsius instead of Fahrenheit, although in our class it really doesn't come up too often. Now, that just means that when we're in the metric system, we're going to be converting between big units and small units. So in our class, we're not actually going to be converting between inches and centimeters, because we're going to always measure things that are already using the metric system. Meaning, when I use my ruler, I'm not using the inch side. I'm going to be using the centimeter and millimeter side. So just as a quick note, if I have something like 0.45 liters, that actually is 450 milliliters. What's really great about the metric system is that it's based off of a unit of 10, which makes converting between different measurements within the metric system very, very simple. If you don't know how to go from 0.45 liters to 450 milliliters, or from 1 kilometer to 1,000 kilometers, that is just not something you know. Again, do not worry. I will get back to you and we will practice metric conversion, which is what this is. So hopefully looking at this slide kind of brings back some memories. King Henry died unusually drinking chocolate milk is what I know a lot of middle school teachers have us remember. And this is a really great reference slide to keep. You have your base unit in the middle underneath the unusually and the crown. And then from there, you can either go bigger or smaller depending on what you're trying to convert to. Okay, so if you need more help, I will make a separate video for this. Now, also keep in mind that there's different uses for this. So for example, if I have liquid, I'm not going to be using mass for that. I'm going to be using volume. If I have something where I need to know the distance between point A and point B, I'm not going to measure its volume. I'm going to use the measurement for length. So there are proper measurements depending on what you need. Now, as I mentioned, this whole chapter, I have at this point 4 more questions you should be able to answer. Number 1, what characteristics do all living things share? Number 2, what are the cross-cutting concepts of biology? Number 3, how do fields of biology differ in their approaches? And I didn't cover this in too much detail, so just make sure you do review that. And number 4, how is the metric system important to science? So if you have any questions, please reach out. I've tried to cover just a portion of the big ideas from chapter 1.